for those who were there on the first part, maybe it is going to be a bit repetitive, but first I have to thanks the MEP Louis Michel. This is thanks to him that we have an access to this room and an access to the European Parliament. And then I also would like to thank our sponsors. You will see them on the screen over there. Okay, so this event has been co-organized by the iGEM UCLouvain team in one side, and on the other side, it has been organized by Young Changemaker. So iGEM, it's an international synthetic biology competition. There are over 300 teams from around the world who present their project in Boston. It's in less than 1 month. And so for this second part, you are going to listen to 4 of those iGEM teams. They will have 3 minutes to present their project. And about our team, because the UCLouvain team co-organized, we are 12 students from 6 different backgrounds. We have medical student, bioengineer, civil engineer, biomedical, and well, we have 6 different faculties in that project. And Young Changemaker, it's an organization who highlight young talents and young people who have impact on their environment. So we decided to organize that event in the European Parliament in order to show that as a student, we can have an impact on things that are generally reserved for big companies or big institutions. Or we wanted to prove that as a student, you can have an impact and you could do something. You don't have to be age, experience, be an academic teacher or whatever. As a student, you could change things. Now it's time for me to invite the first team. So Alex, you have 3 minutes to present the project, and I have the timer somewhere, but okay, over there.
So hi everybody. So I'm Alex from the iGEM team from UCLouvain 2018, and I'm gonna do a quick presentation and overview of our project. So that's called Backwards. So 2 years ago, the World Health Organization made sound an alarm for the awareness on multi-resistant bacteria, and that will impact on the human health. So based on this event, we developed a biological tool that will be able to neutralize infectious bacteria. And so this tool was based on two smaller biological tools. The first one is conjugation. So conjugation is an ability of bacteria to replicate and transfer its DNA in the form of a plasmid to another bacteria, and in the end, the two bacteria will share the same plasmid with the same information. The other tool is CRISPR-Cas9 that was presented earlier. So CRISPR-Cas9 is a protein that can cut a double strand of DNA with the help of a small guide that's formed from RNA. In our project, we use this guide for RFP, so red fluorescent protein, and for ampicillin resistance gene. And so when we combine the two of them, we have a donor cell with our plasmid with the Cas9 and the guide gene. So the transfer of the plasmid occurs to the recipient cell. Then the plasmid is expressed, and there is production of the Cas9 and the guide RNA that will go cut the unwanted gene in the recipient cell. Of course, if there is no unwanted gene in the cell, in the recipient cell, there is no cut and the cell keeps living. So first, we wanted to try if our Cas9 in our conjugative plasmid was still active and could work. So our control is a strain that produced RFP, so red fluorescent protein. That's the little red dots that you see. And on the right side, you see the same strain where we added our plasmid, and we can see that there is no more red dots, so the RFP gene has been cut. So that proves that our Cas9 is working with his guide, and and has an efficiency close to 100%. The second, we wanted to prove that the conjugation was still working. So in blue on this image, you have the donor. On red, you have the receiver that produced the RFP still. And when you conjugate both of them, you obtain a white colony because the RFP gene has been cut. So what are our perspectives for the last month of this competition is we want to have more quantitative data for this project. We want to try the conjugation with other strains. We have collaborated with hospitals to eventually try it on Vibrio. And last point, we want to do a time lapse under a microscope to for more visualization of the conjugation phenomenon of our project. Thank you for your attention.
Are there any questions for our project? So then I'd like to invite the Paris team. So it was over 3 minutes, but whatever. Yeah, actually the iPad screen stopped, so I would say it's my fault. So okay. So I will give you the microphone.
Hello. It will not be the same kind of presentation. We don't have PowerPoint. Just to let you know. So maybe I can introduce you who we are. So we are. It's not part of the presentation. You were just to say something between the time. So I will just keep on doing that. We are a team composed of 20 students from diverse schools in Paris, and this year we are working on the project Neuron Arch. So should I start? Yes. Okay. Thanks. Sorry. Can you see this gesture? It may seem instinctive to us to do this, but if we have to think about, we just do it. We don't have to think about how to do it. But if we start thinking about how we do it, it may seem a little bit more complicated to just grab this bottle. And it is. For a lot of people in the world, it is a real challenge. Amputees, due to their conditions, they are not able to perform daily life gestures. This is why upper and lower limb prostheses mimic amputees missing members and giving them an aesthetical or functional aspect. And there is some bionic prostheses, so the most performant ones that can allow amputees to recover a partial autonomy and accomplish simple gestures, so just grab this bottle. But it is done mainly with nerve connection, nerve redirections, and muscular contractions. However, this technology does not involve the direct connection between the nerves and the machine. And this is the key point for future prostheses, for a better autonomy. This is where we have a role to play. In our project, Neuron Arch, we are using synthetic biology. So we are using synthetic biology, and we are creating an interface that allows the nerve connection to an electrical implanted devices. So for that, we use a community of microorganisms, a biofilm, that will allow the direct nerve growth to an electrical implanted. So this biofilm is secreted nerve growth factors. The nerve growth factor is a protein capable of induce the nerve growth toward our interface. And all devices will be able also to amplify and transmit the signal to the mechanism of control of the prosthesis. But moreover, we also want to tackle another burden that is regarding orthopedic surgical infections. So these infections, they are mainly due to the formation of pathogenic biofilms on the implanted device in 5% of the case. So these biofilms, they are very resistant to antibiotic treatments, and the last way to remove the infections is the reoperations. These have an heavy and costly cost, and it is heavy and costly for both the patient and our health system. So this is why with a biological approach, we want to tighten up the gap between actual prosthesis and the unmet needs of amputees. Thank you.
Does someone have a question for the very team?
Ouais, je préfère aussi. En fait, on développe une interface, donc notre biofilm, c'est l'interface qu'on viendrait poser au niveau d'un membre amputé pour justement permettre la reconnexion des nerfs avec un implant électrique qui permettrait de relayer le signal nerveux. Merci. C'est mieux en français aussi. Oui, alors ça sera sur les nerfs du système périphérique. D'accord. Alors en fait, on voudrait être ostéo-intégré, c'est-à-dire directement placer une branche de titane au niveau du moignon amputé. Et sur cette branche, il y aurait toute une partie où on viendrait poser notre biofilm qui serait contenu dans une membrane elle-même conductrice aussi. Est-ce que ça répond à votre question? Ok.
Thank you very much. So I propose we continue in English because, first of all, there are people from Sweden today, from the north, from Holland, and I did not told that before because I did not want to add more stress. But we are actually live right now, and there are over 300 people watching the event right now. I just got the confirmation. So and those people, they are from many other places because iGEM has 300 teams, and so we have people from around the world, exactly more than 300 people watching at the event. So now I would like to invite the Eindhoven team to present their project. And so I give you the mic. Okay, I'll just give you the mic. Okay.
Thank you. We are a team from Eindhoven, the Netherlands. As you can see, we are a very beautiful team from Eindhoven with 12 people, interdisciplinary team from the Technical University of Eindhoven. And we have come up with a living biomaterial. What is a living biomaterial? A living biomaterial is a biomaterial in which cells live that cooperate to achieve something or interact with the environment. The only problem about living biomaterials that are very much wanted in the biomedical field is that bacteria and living cells in the biomaterial escape and migrate out. And this brings some risks and health risks with them, because you don't want genetically engineered organisms leaking out of a material and escaping into the environment. That's why we came up with a solution. We found a bacteria, an Arctic bacteria, that secretes a domain that binds to sugar. It's a sugar binding domain. And we transformed this bacteria into E. coli, and it works as an anchor. It grows out of the bacteria and it binds to sugar groups. So we synthesized a gel out of dextran monomers, and that's a sugar polymer. And we synthesized dextran hydrogel, and the sugar binding domain that sticks out of the E. coli binds to the dextran monomer and polymers, so that the E. coli bacteria are stuck in their place inside of the gel, so that they can't escape and function and interact with the environment outside of the hydrogel. So we have created a living material that is safe to use outside of the lab, and the applications that you can use it for are endless. You can use it for drug synthesis, you can use it for biosensing, energy creating. Yeah, the world of synthetic biology has a lot of options. And we chose to go further in the application of therapeutics. So we're letting the bacteria inside the living biomaterial synthesize lysostefin, which specifically attacks and kills Staphylococcus aureus. And that's a bacteria that grows in infected wounds, and that's one of the most hard-to-kill bacteria in infection wounds. And with that, we create a patch that you can apply to burn wounds or other infected wounds, and thereby giving a continuous supply created by the bacteria in a living biomaterial to fight this bacteria and to make the wound bacteria-free without the use of antibiotics. So yeah, that's our presentation. Do you have any questions? Yes. What do you mean? On a real patient? Or no, we didn't do that yet. There are some restrictions that so we can't use it yet on other patients. But we got the bacteria stuck in the hydrogel, so it binds to the hydrogel we synthesized, and we got an excretion program for lysostefin. So for the protein we want to secrete out of the hydrogel, and yeah, it seems that it's going to work. And we need the last results yet, so we're finishing up the last transformations. But I think you will hear more about it in a few weeks on the BOS in January. So, any other questions? Okay, thank you.
Thank you very much. So I'm going to invite the team from Sweden, from Lund University. They came this morning from Sweden to came to that to this event. So just when you ask a question, please open the microphone on the table, because otherwise the live video does not go through. But that was the small information. Okay, so let's go.
Before I started to talk about the project, I just want to say that I'm very grateful to be here today, and it's so nice to see so many students here on a Saturday. You could have done something completely different, but you decided to come here and listen to this. So our project is a manufacturing project, a biomanufacturing project. So that means that we focus on creating different products using microorganisms. So in bioprocessing, you usually have a fermenter tank, as you can see here. You have microorganisms plus nutrients, and they react and they form different products like pharmaceuticals, vaccines, enzymes, detergents, things you use in your everyday life. But there are some issues with this, and those are different bottlenecks. Sometimes you have low product yields, you have a very high energy consumption, and mainly there are oxygen limitations because these organisms, they need oxygen to procreate and produce the targeted product. So what we are working with is that we are using a very special kind of hemoglobin from an organism called Vitrocilla to increase protein production. And how this hemoglobin works basically is when the organism is in a very oxygen-poor environment, it starts to produce this hemoglobin, and it acts almost kind of like a battery. So it can continue to grow even during low oxygen concentrations. And this hemoglobin has been used in various applications, for example, bioremediation, for example, degrading textile dyes. And they've done it in one study and improved it by 20%. We can use it in plants, increase the dry weight in maize, for example, and it also improved drought resistance. So, for example, when you have floods, which basically suffocates the roots of the plants, it increases the resistance to that as well. And also in biomanufacturing, production of amylase, galoxidases, which are used a lot in the industry. So how we do it is that, our approach to it, like this has been done before, but what we want to do differently is to see at what degree the hemoglobin expression, like how that affects the protein production. Because if you, if you express too little of it, then you won't have any effect on the cell. And if you express too much of it, all the cell resources will go to producing that hemoglobin. So the answer lays somewhere in between, and that is what we want to try out. We design a couple of different constructions, and you can see that the promoter strength increases. We have different promoters for each construct. And we use GFP as our proof of concept protein. So we measure that intensity at different promoter strengths. And the hypothesis is that, as I explained before, that we should get somewhere in between there. And this is our results. As you can see, this is preliminary data. We're still testing this out. But as you can see, there is an increase in green fluorescent protein intensity as the promoter strength increases. So the next step for us is to try it out with an industrially relevant protein. And in our case, we've decided to go with the Z domain of protein A. I'm not going to go into that, but it's used to look for different antibodies. So basically, we're trying to create an universal solution in all bioprocessing scenarios. And basically, ideally, you would have your target protein and you would add the hemoglobin to it, and then you start expressing it and hope that the yield increases. So our project has a strong scientific basis. There's a lot of studies done on this, and it works. It works in various host organisms: bacteria, plants, yeast, higher eukaryotes. It adds an additional degree of freedom. So in biomanufacturing now, you have a way to control for the oxygen content. You can change that according to your needs. And it's a novel synthetic biology approach. You can basically add whatever you want to it, whichever protein. So thank you, and I'll gladly take your questions. Yes.
What is for you the main application of your project you're working on?
The main application, it is for bioprocessing. So when you have microorganisms producing some kind of product, you want to tailor the oxygen demand for that microorganism. So this gives, as I said, an additional degree of freedom. So we want to make more oxygen available for the cell.
Thank you.
Yes.
I'm just wondering because in an organism like humans, for example, the blood is circulating, so the hemoglobin takes oxygen in the lungs and then delivers it to the muscles and take CO2 back.
Yep.
And I think hemoglobin has a much higher affinity for CO2. So how exactly is the hemoglobin protein going to not take the CO2 produced by the bacteria in the reactor and instead take O2 and deliver it to the bacteria?
That's a very good question. This is not human hemoglobin. This is Vitricilla hemoglobin. So it acts in different ways. And what makes it so useful in this case is that it binds oxygen very strongly, and it releases it quite easily. So that's why it has so many applications, and that's why it can. There are a bunch of details that I could go into, like how it works, but it acts different compared to human hemoglobin. Yeah. All right, thanks.
Thank you very much. So I'm very glad that all the teams were able to present their project. I think it was a little technical, but I'm not sure my grandmother would have understand the whole presentations. But still, I'm very glad that everybody were able to present their ideas.